thank you for being here now for the Clinical Trials Showcase. You've heard a lot about HD science and research over the last 2 days, but now you're going to hear about some opportunities to get involved in currently enrolling and soon to enroll clinical trials. You've heard us talk about how important participation is at the research forum earlier this morning, and I want to emphasize that again now. I know that taking part in a clinical trial can be a big commitment, and I encourage anyone who is considering one to make sure you understand what it is that you are signing up for as best you can. I also want to say something that might sound kind of blunt, but it is a real truth. Without participants, clinical trials can't happen. Without clinical trials, potential treatments can't be tested, measured, and hopefully make it to clinic. As we've learned over the years, even if a trial does not meet its expected endpoints, please know that everything that is learned in that trial is valuable and helps inform what happens next. I hope that while listening to our speakers today, you will hear about a trial you might want to participate in, or maybe even tell a family member who wants to participate that isn't here right now. We also want to learn from you during this time. While you're enjoying your lunch and while you're listening to the speakers today, please take a minute to scan the QR code that's on your table with your smartphone and complete a survey. This survey is going to help HDSA better understand clinical trial readiness in Huntington's disease. We can't move forward with clinical trials and drug development without your participation, and we want to better understand your questions, concerns, and hesitations so we can address them and work with our corporate partners so that they can do that as well. Again, we're going to have some speakers today talk about currently enrolling or soon to enroll clinical trials. And we have 5 people speaking today, so it's going to be a packed session. Each person is going to have 10 minutes with about 5 minutes of Q&A. Now, just to preempt this, I know that there's a lot of information to be shared. So if our speakers do run over and they're not able to take as much Q&A as possible, please know that they are all here through the rest of the day out in the exhibit hall. If you don't get a chance to ask your question, please find them and ask them at that point. It is also a packed schedule, so speakers, I found a cowbell. And I'll be the one standing here at the edge of the stage. And this thing just jingles and makes a lot of noise as I'm holding it. But just imagine what it does if you're running over time. So I'll let you know. All joking aside, I'm super excited for our speakers to talk to you today, to tell you about what is going on in current clinical trials that are enrolling or soon to enroll, and really hope that those of you listening consider which trial you might want to participate in and get more information about that. And please do share this with family members who aren't in the room, because we know not everyone could be here today, and there's opportunities available for all. So without any further ado, please help me welcoming our first speaker from Wave Life Sciences. Here we go.
Good morning, or good afternoon. My name is Jane Atkins, and I am the current program lead for our WV003 program, and I wanted to thank HDSA and the organizers for giving me the chance today to talk to you about 003 and Wave's attempt to do our best to take a whack at mutant Huntington. So the QR code on this slide will take you to our full disclosure statement, the gist of which is basically to say that we're a science and research-based company, and inherent in that research is a lot of uncertainty. We have a lot of hope, but there's still a lot of work to realize that hope, and because of that, I'm going to be using words like may or might to reflect that uncertainty, and I wanted to note that those are only predictions and not facts. So for those of you who are not familiar with Wave, we're a biotechnology company working to bring transformational therapies to those with serious or life-altering diseases. We're located just outside of Boston, and we've been actually working on Huntington's disease for over a decade. As shown on this slide, Wave was formed in 2013, and unfortunately our first attempt with our Precision HD program did not work, and we discontinued that program in 2021. However, in September of that same year, we initiated Select HD, which was a trial to look at our second-generation molecule, WV003. We had positive interim results in 2022, and we read out the final results of the study in 2024, last year, and I'm going to be going over the key results from that study today. Before I do that, though, I wanted to take some time to go over WAVE's rationale for how we're tackling HD. The key point in our approach is that we are looking to selectively lower the mutant Huntington protein while preserving and not touching the wild-type protein as much as possible. There are a few key principles that underline that rationale, the first of which is that HD is caused by mutant Huntington. I want to thank Rachel for going over a lot of this morning, so I don't have to go over this in detail. But recall that in healthy individuals, they have a copy of 2 healthy Huntington genes, and in individuals with HD, they have 1 mutant or abnormal copy and 1 healthy copy. It's this abnormal copy that makes mutant Huntington. And the literature and the studies have shown that HD is caused by a combination of effects caused by the presence of the mutant Huntington, but also by the fact that there's less wild-type Huntington. So wild-type Huntington has been shown to be important for a number of critical brain functions, having to do with communication as well as the health and survival of brain cells. In addition, there are studies that have shown that wild-type Huntington plays a critical role in the flow of fluid, or cerebrospinal fluid, within the brain regions. And this is important because it's this flow of CSF that allows brain cells to get their nutrients and also to get rid of their waste. Another key principle underlying WAVE's rationale is the concept of a SNP, or a single nucleotide polymorphism. So SNPs are normally occurring variations in the DNA. They're located on specific spots, and they're single letter differences, and they are often associated with specific genes. So because of this, if you can identify the right SNP, you can potentially identify a specific gene. So WV003, WAVE's oligonucleotide, looks for a SNP that we call SNP3, and we found that this SNP is associated more often than not with the mutant Huntington allele. So by targeting this SNP, we can target just the mutant allele and leave the wild type relatively untouched. Literature suggests that about 40% of the HD population are going to have SNP3 on the mutant allele. So now I'm going to go into the results of SELECT-HD, our first-in-human study where we looked at 003 in HD. SELECT-HD was an adaptive first-in-human study that was completed in June of last year. It was a multi-center randomized double-blind placebo-controlled study that enrolled both males and females ages 25 to 60 who had early manifest HD, and importantly also had that SNP3 on the mutant allele. The primary objective of the study was to look at the safety and tolerability of 003. But we also wanted to get a handle on how 003 was distributed in the body, and we wanted to get an initial sense of whether it was working or not. And so we looked at levels of mutant Huntington protein as well as wild type or healthy protein in the CSF. And we also looked at levels of NFL, which Rachel also talked about this morning. In addition, this was a small study. We knew that we wouldn't get really robust data, but we did also look at clinical assessments and exploratory MRI or imaging of the brain in this study. So the results are summarized here, the key points. So the first thing is that we found that 003 lowers mutant Huntington protein in the CSF. That's exactly what we wanted to see. Yay. Secondly, we confirmed that we were leaving the wild type protein relatively untouched. So again, very important, confirms that it is working as we designed it to work. We were extremely happy. We also saw for the first time an association between the lowering of mutant Huntington protein and the slowing of caudate atrophy in a clinical trial. And I'm going to be talking more about caudate atrophy and explaining that a little bit more in the coming slides. So I know those are words that not everybody understands what they mean. We showed that 30 mg of 003 when it was dosed 3 times, given once every 8 weeks, was generally safe and well tolerated. We did see some elevations in neurofilament light, or NFL, but importantly, when we looked at these very closely, we didn't see any correlation with the number or severity of adverse events in the study, and we also didn't see any correlation with changes in the caudate volume. So the general, and the last point I want to make is that when we looked at the data and we did some modeling, we believe we are planning actually to dose in the next study every 12 weeks rather than 8 weeks, which is what we do. So that's a more convenient dosing frequency, less times in a year. The QR code on this slide will take you to a webinar, a Breaking Down Barriers webinar with Wave, where we actually go into the details of the study results in more detail if anybody's interested. So here's a key result from the study. This is a graph that shows the amount of mutant Huntington in the CSF over time. So along the bottom of this graph are the arrows that shows when 003 was given. It was given 3 times every 8 weeks. The blue line is the amount of mutant Huntington in CSF for those patients treated with 003, and the gray line is placebo. And you can see that by day 169, we had a maximum amount of lowering compared to placebo of 46%. And this was really durable because at day 197, it was still down. And it's this data that gives us confidence that we can dose every 12 weeks in our next study. We then also looked at wild type, or the normal healthy protein. And again, you can see the treated patients, or the treated individuals in the blue line, their levels of the normal wild type Huntington never went below those of the placebo, statistically speaking, given by the gray line. So these two results together confirm for us that 003 is working as it's intended. It's selectively lower the mutant Huntington, and it's leaving the wild type relatively intact. This slide summarizes the safety of 003 given 3 times. It was generally safe and well tolerated. All of the adverse events were mild to moderate intensity. We didn't have any SAEs or severe adverse events at the time the study ended. So before I go on, I want to talk a little bit about what a biomarker is. Again, Rachel did me a favor. She went over this already this morning. A biomarker is basically a measurement that can be used to see how well the body responds to a treatment. Mutant Huntington in CSF, wild type Huntington, NFL, those are all biomarkers. These are all biomarkers that give us a sense of how the treatment might be responding, or how the body might be responding to the treatment. One thing that researchers are always looking for are biomarkers that give us a hint of how well a treatment might be affecting disease progression. So is there a biomarker that kind of correlates or goes along and shows us how Huntington's disease is progressing? At this point, there's been a lot of shout outs already, but I want to give it another shout out of the importance of observational and natural history data. It's because of this data that we're able to go and look, and what's been shown, a key thing that's been shown is that one of the earliest changes that happen in disease progression in Huntington's is that brain cells in a region of the brain called the caudate start to die. Right? And it's that data that, because of this, we went and in Select HD, we looked to see how we were potentially impact the size of the caudate, which is that deep region of the brain, and whether we were potentially having an impact on how fast the brain cells in the caudate might be going away. So what we saw for the first time is that we saw a correlation or an association between the amount of mutant Huntington lowering and the slowing of caudate atrophy, or the loss of brain cells. So I've looked at this slide multiple times, and I've said this before. To somebody who is not used to looking at data, it looks like a bunch of dots, and you just drew a line through it. But from a mathematical statistical point of view, we drew that line and we did some statistics on it, and those are the numbers that you see up in the upper corner of the graph. The R value gives you a sense of how good that correlation is. Anything close to 1, the closer to 1, the better. So the number here is about 0.5. That's not bad. It's not great, but it's not bad. And this was very preliminary data, not a lot of dots. We need to do this again and get a lot more dots on there to confirm this finding. Right, but basically what this means is that the more mutant Huntington lowering you have, it looks like that you're losing less of the caudate, right? And so an example here is shown that over on the far right, if you have the least amount of caudate lowering, that equates to about a change of about 8 cubic millimeters of caudate. However, if you have about 40% mutant Huntington lowering, it looks like it's only about 4 millimeters cube of caudate. So this is really encouraging, but I also want to emphasize very preliminary, we need to do it again. We need to do it again with a lot more patients to confirm it and to see if it actually does lead to a slowing of disease progression. Still very preliminary. So next steps now is that we are actively working to develop our phase 2/3 study design, which we hope will be a potentially registrational study in adults with SNP3 and HD. We're going to be looking at caudate as a primary endpoint in this next study, and our plan is to submit this IND in the second half of this year, which I guess the second half starts Tuesday, but but that's our plan. And again, I guess I can't emphasize this enough. I know it's been said, but we are all over the natural history data as we're planning this study. So all of you guys who have participated and have enroll, as well as older studies like Track and Predict, that data is invaluable to us right now as we're planning this study. So thank you. And again, thank you all, and as well as the investigators and the staff and the entire HD community.
Jane, thank you so much. I didn't realize there was a counter here, so I'm sorry for lurking off the stage earlier. But we've got a couple minutes for questions, if anyone has a question for Jane. I see a question over here if we've got folks with mics, and I see Nakia sprinting. And he sprinted too far.
It was a wonderful talk. What are your very next plans now in your research? And it's all, for me, it's all about the hope that gives me encouragement.
Yeah, so as I said, our very next plans are to do another study. We're planning a study that's going to enroll about 150 patients. We are planning to enroll patients that are a little bit earlier in disease, HDISS1 and HDISS2, and hopefully that's going to give us data to show that 003 can help slow HD, and hopefully we'll go from there.
Thank you.
Thank you. Do we have one more question that we can offer to Jane? Y'all just want me to play the bell, right? Like that's...
Yeah.
If not, please know that you can catch Jane and her colleagues out here at the Wave Life Sciences booth. Thank you so much for that. Totally appreciate it. All right, next up, I'm going to invite Lori to the stage to talk about Mend HD.
Perfect.
And you'll have a timer there, so you don't need to worry about it.
Very good. Hi, everyone. It's nice to be here. My name is Lori Quinn. I am a physical therapist and a professor of movement science and kinesiology at Teachers College, Columbia University. So I'm here to talk to you about a new observational study that we are running called Mend HD, so digital measures for clinical trial endpoints in Huntington's disease. So Mend HD is an FDA-funded observational study, so that means it really just happens right now at one time point. We're hoping to have it be a longitudinal study. And we're assessing the feasibility of using wearable technologies. So wearable technologies are those technologies that are on your body, so typically wrist-worn devices like an Apple Watch or a Fitbit, that type of thing. But we're also going to be having devices worn on the trunk or on the back. And we're going to be assessing the feasibility of using these types of technology to passively record outcome data. So this means nothing needs to be done by the participant. They just wear the device and the data gets passively collected. Sounds like a good deal. Specifically, we're going to be looking at measures of gait or walking and chorea, which are the involuntary movements. And our hope is that these will be able to be used as clinical trial endpoints in people with early stage HD. And as you've heard from WAVE, and I'm sure you'll be hearing in some of the subsequent studies, a lot of the research is moving to early stage, but we also really importantly need clinical trial endpoints that are sensitive to the problems that people have very early in the disease. So we're really hopeful that wearable sensors, like the ones we're going to be using, can be used to help in clinical trials and can be actually used as primary outcomes. So our long-term objective is to establish these digital measures as treatment response endpoints in early HD in line with FDA guidance. And the really important thing about this study is that it's actually funded by the FDA. So we're hoping that will really help to fast-track the approval of these endpoints. So why are we running the study? Well, our study will help to validate the digital measures of gait and chorea, which we think are more sensitive than the traditional rating scales that are currently used. We'll also evaluate other measures, including daily living physical activity, heart rate variability, and sleep quality. And really importantly, we're not just going to be collecting these digital measures on their own, but we're going to be linking them to participant interviews. So we're going to be having very detailed interviews with the people in the study, as well as their care partners or family members, to try to understand exactly which aspects of gait and chorea are bothersome, and can we link those to the digital measures. This is something that's really important for the FDA. So who can participate? And we are just starting this study, so it's actually perfect timing. We just started enrollment last week. So all participants must be between 25 and 60 years of age. We might increase that to maybe 18 to 65, but right now we're starting with somewhat of a narrow window. And participants must be diagnosed with HD, and it must be genetically confirmed and in the early stages. If you think you might qualify, we're definitely going to encourage you, even if you're like, I'm not sure if I'm early or if I'm mid, we really encourage you to sign up, and then you can meet with a study coordinator who can do the screening. I know this is a busy slide, but this just goes through the outline. It's just a quick timeline that goes from left to right. There's 4 visits, and the beauty of this study is it's completely remote. So you can be anywhere in the country, not in the world yet, but anywhere in the country, and participate in the study. You will be meeting with a neurologist and a study coordinator remotely via Zoom to do all of the intake. We're going to be doing the virtual UHDRS. Some of you may have heard of that, which is a remote rating scale specific for Huntington's disease. And then we do some baseline assessments. That's visit 3, where we have people move around and we watch them on telehealth. And then we give people 2 devices. One is worn on the wrist, and one is worn on the back. And those you wear for about 2 weeks. Again, just going about your daily life. You can take a shower with them. We wouldn't expect any deep sea diving or anything, but you could probably go for a swim with them. They're all waterproof. You really should just pretend that they're not even there. And then you'll mail them back to us. So we mail you everything, and then you mail them back to us. And then the last thing will be the qualitative interview, that sort of in-depth assessment that I talked about. So some of the study highlights are that you can participate from the comfort of your own home. It'll be really nice with really lovely neurologists and study coordinators who will meet with you via telehealth. And so there'll be 4 visits. We'll mail you all the study materials, so everything will come right to you, and you don't have to travel anywhere. And the sensors that you're going to be wearing are relatively small and non-obtrusive. If you have a watch or something that you currently wear, you could just wear this device on the opposite side. So it should be relatively easy. And maybe most importantly, you get paid $200 for completing the last visit. So what are we measuring in this study? Well, I talked about gait and chorea. Specifically within gait, we're looking at gait quality, so the quality of someone's movement. Some of the really subtle movements that we think we can be picked up by the sensors are things like variability of movement, but also gait quantity, basically how much someone walks in a given day. The sensors are sensitive enough to pick up very subtle choreic movements, which can come early in the disease, so we'll be looking at that. And then various measures of physical activity and sleep. So time spent in sedentary behavior, total physical activity, time in bed, total sleep time, measures of sleep efficiency. These are things that have been measures, but typically in like long-term sleep studies, which require people to go into a clinic, this will be one of the first studies that'll really be validating it with these simple sensors in the home. And then finally, measures of vital signs like heart rate variability. Okay, so our timeline, here we are, June 2025, and we are just starting recruitment, so it's a perfect time. We've already had people come and sign up. We have a table right across from this hall. We hope to do our first screenings next month, very soon, and then we'll be collecting data and doing all of the assessments over the next 9 to 10 months. Then we'll work on data cleaning through June and August of next summer, and then finally completing data analysis. What are the outputs from this study? Well, we hope to determine the sensitivity of these sensors to detect disease severity and ultimately disease progression. Right now, as I said, this is an observational study at one time point. If there's any funders in the room, we would love to get additional money to be able to continue this for over multiple years. You heard previously the importance of these longitudinal studies like Track HD and Enroll HD that are longitudinal. We would like to be able to do the same kinds of assessments with these wearable sensors over longer periods of time so we can see how sensitive these are as the disease progresses. We also want to be able to map these digital measures to meaningful aspects of health, so really what matters most to people with HD and their family members. Third, and I think this is really critical, because this is funded by the FDA, we are required and we want to provide open source algorithms of gait and chorea. So anyone will be able to use the data and use the algorithms. So any companies out there that are developing new measures for new interventions for Huntington's disease, they will be able to incorporate these outcomes. And then finally, providing a roadmap for how digital measures will be used in clinical trial settings in HD. So this is our study team. Jamie Adams, who is a movement disorders neurologist at the University of Rochester, is leading our team, along with myself and Jeff Hausdorff, who is an engineer at the Tel Aviv University, and Jennifer Mammon, who is leading the qualitative assessments, and she's at the University of Massachusetts. And we have a whole bunch of really great research assistants and associates as well. So I'll leave this up. Hopefully everybody's going to get their phone out and scan the QR code. I also have plenty of flyers outside, so I hope that some of you will plan to join up or to share it with other people. Again, I think the remote aspect of this study is really unique, and it can really help not just one intervention, but possibly many or all of the interventions that are coming down the road in HD. So we're really excited about this study. And finally, I just wanted to put in a plug for the HD Research Room. I've seen many people here who've already come to the research room. I'm a physical therapist, and we have also been doing some balance assessments there. We've already seen over 100 people, so I just want to say a huge shout out to everybody, because that is an incredible turnout. And I've been doing research in HD for a long time on physical activity and movement, and I've never had a study where I had that many people, and it usually takes me many, many months, if not years, to get 20 or maybe 25 people in a study. So we're really grateful. Everybody who is participating, it's incredibly helpful in advancing the science very quickly. So please come, we're still open until 5 o'clock. Thank you very much.
See if we've got some questions for you.
Sure, absolutely.
I just want to emphasize something that Lori said, and we've talked about this too, is just the power of the people who participate in research. And the fact that that many people took part in this over the course of 2.5 days. I mean, you still got time, 5 o'clock today, but any questions for Lori about the MEND HD study? All right, we've got one over here, and then we'll go over there next.
Hi, I had a question about the device.
Where? There.
Right here.
Thank you. Oh my gosh.
So
this
the
is crazy.
device, does it pick up movements like all over the entire body?
Yeah, this is an excellent question, and the FDA just asked us that the other day. So it will pick the measurement of the trunk and the wrist. If someone has movement of the wrist, it will obviously pick it up. But if you had movement of, let's say, the wrist that the device wasn't on, even a subtle movement of the wrist, you get a very subtle movement in your back, unless you're very good at stabilizing. Or if you have movement of the leg, it should also be registered in the trunk. But this is something that we're going to be looking at. In the study that we're doing here, we have sensors on 6 parts of the body, which is more sensitive for sure. But the problem is that's really cumbersome to go about your daily life wearing 6 sensors on your body. So we are trying to balance out the feasibility with what we think is the best chance to get that information.
The reason I'm saying that, thank you for answering that. I have movements in the toes and fingers that are a lot, like really, that's how it started. So, and they still continue. But I was just wondering if it picked up from there.
It's a great question. I think the toes are going to be tricky. So if somebody just had chorea just in the toes, I don't think we're going to get it with that. Okay. We will get that from the questionnaires, though, and the information. So it'll be interesting to say if people are reporting that they've got it in the toes, but our sensors are not able to pick it up. But certainly in the fingers, we already know that we can pick it up. We can pick up the subtle movements of the fingers because you get a small acceleration change in the wrist sensor. Thank you.
Thank you.
Yeah.
You mentioned that you have an engineer on your team. I know someone who's a mechanical engineer. Is that a field that he could consider?
Oh, I love this question. Yeah, I mean, I think there's many different kinds of engineers, and so I think, sure, and you can send him over to us. The engineer that we're working with, Jeff Hausdorff, is more on the computer engineering side of things, but I think anybody who is interested in doing this kind of work and doing these analyses, I'm sure Jeff would be happy to talk to him.
Thank you.
Yeah, great question.
I like that she's actively recruiting.
I know, thank you, yeah.
More over there.
My question is, so it's obvious that if people participate in this study, that all the information goes for research. But does that participant get any feedback, any kind of, not diagnosis, but any kind of?
Yeah, so we are definitely planning to do that. And one thing that I didn't mention is we have a wonderful patient advisory group who's advised us along the way, and that's something that they mentioned is really important, is sharing individual data back with participants. So that is something that we will be doing about their, especially because a lot of the information on wrist-worn devices are things that you might be getting if you had an Apple Watch or a Fitbit on, and we'll be sharing that information in a report back to participants. Yeah, thank you. Good question. Okay, anybody else? Oh, there's one more.
Oh, one more.
Oh, somebody's coming. I
run
don't think
left
that's
and right.
allowed.
Thank you. So I think on one of the slides it said you're looking for people to volunteer, 20 people who are not impacted by
Yeah,
HD?
so
Is
we
that-
do need healthy controls, control
Okay,
participants.
and that can be anybody, anywhere?
Anybody, anywhere. Still between 25 and 60.
Great, thank you.
Yeah, please do sign up. It's really, I think will be fun, and we really could use the help, so thank you. Okay, thank you.
Thanks, Lori. All right, next up, Rita, come on up. Thank you.
Thank you. So press this backwards. Good afternoon, everyone, and thank you for inviting me today to speak. My name is Rita Gandhi, and I'm a Senior Medical Director at Genentech, part of the Roche Group. Today, I'll share where we are, where we're going, and on the road towards disease-modifying treatments for Huntington's disease. So just a quick reminder, the programs I'll mention are all investigational. They're still being studied and are not approved for routine medical use. And nothing in this talk is personal medical advice. So please keep working with your own healthcare team for decisions about your care. And again, I'm an employee of Genentech, a member of the Roche Group. For over a decade, Roche, Genentech, and our partners have been deeply committed to working alongside scientists, clinicians, and most importantly, families within the Huntington's disease community. Through this collaboration, we've gained valuable insights into HD, and just as importantly, a clear understanding that there is still much to discover. We know that mutant Huntington protein causes HD, making it a critical target for therapeutic development. However, as many of you know firsthand, drug development is rarely straightforward. There's no one-size-fits-all solution, no single medicine that will meet the needs of every individual at every stage in Huntington's disease. That's why I'm proud to share how we are now advancing multiple research programs in parallel. By taking this multi-pronged approach, we aim to deliver safe, effective, and meaningful treatments for HD as quickly as possible. We're pursuing 3 different investigational approaches that aim to lower the Huntington protein, and I'll give you a brief overview of each, which are all in different stages of research. Tominersen, many of you are familiar with, is our most advanced program. It was the first drug to show that mutant Huntington could be lowered. This drug is well into the phase 2 study and had an important update in April. I will formally introduce RG662, our gene therapy candidate, as well as a new drug candidate designed to selectively lower mutant Huntington protein. So let's go into tominersen. Tominersen is an antisense oligonucleotide. Think of it as a short piece of genetic Velcro that sticks to the message telling cells to make Huntington protein and then marks it for cleanup. Currently in phase 2, tominersen is designed to lower both types of Huntington protein. It's the focus of our Generation HD2 trial, which is fully recruited and actively underway. From previous trials, we know that tominersen can lower mutant Huntington, but we need to know if that translates into functional benefit. Tominersen was being studied in a large phase 3 trial, which unfortunately had to halt dosing early. However, upon analysis after the study, we saw that a subset of people, those with less progressive disease, and those who received lower doses of tominersen may have seen benefit. Therefore, Generation HD2 aims to answer an important question. Can tominersen be beneficial if given at lower doses to people with earlier HD? This study aims to answer that by looking at safety, biomarkers, and signs of clinical benefit. And until recently, the study was testing 2 dose levels of tominersen. An important update happened in April this year. As with most clinical trials, there's an independent group of experts monitoring how a study runs. They regularly review data to ensure participant safety and give recommendations to study sponsors to continue, modify, or stop the study. In April, we changed the study following recommendations from the IDMC. So what did they say? Most importantly, they recommended the study continue, and they said they had no concerns about safety. Then they told us that one of the doses was more likely than the other to potentially offer clinical benefit, and therefore recommended we stop giving the participants 60 milligram dose and move those people to a 100 milligram dose. The study is ongoing, and we look forward to data results in the not-so-distant future. Here's a visual look of the updated study and the different testing groups. The vertical line represents the changes since the IDMC recommendation. The study continues to collect data. All participants continue to receive lumbar punctures 3 times a year. Dose switching was done in a blinded fashion, meaning treatment assignments are not known by the study doctors, the participants, nor Roche. In summary, we are incredibly grateful to the 301 participants and their companions enrolled in Generation HD2 around the world. Each study visit contributes to collecting data that helps understanding about tominersen and the Huntington lowering strategies. The IDMC will continue to regularly review data every 4 to 6 months and make recommendations to continue, modify, or stop the study. And since the study is fully recruited, we are now in the final lap, and we can say that data are expected next year. So stay tuned, and thank you for this community's ongoing commitment to this program. Now, I'd like to shift gears and revisit some of our early research efforts. Some of you may already be familiar with Spark Therapeutics, a leader in gene therapy, and for the past several years, part of the Roche group. This year, Spark was fully integrated into Roche, and with that transition, its Huntington's disease program has officially become part of our portfolio. Gene therapy aims to transform genes into medicines for people with inherited diseases, offering the potential for long-lasting impact from a single treatment. Like tominersen, RG662 targets total Huntington protein, but as I mentioned earlier, we believe there won't be a single path forward. So let me walk you through the unique approach behind this particular therapy. The goal of gene therapy in HD is to deliver genetic instructions to stop the production of Huntington protein by delivering the medicines directly to the parts of the brain that are most affected by HD. This includes a caudate, the putamen, which are in the central core of the brain. A specialized neurosurgery is needed, but the hope is that a long-lasting benefit can be achieved with this one-time procedure. For several years, our Spark colleagues have been engineering this gene therapy on their specialized platform and completing the necessary animal testing. This year, we were able to start our first in-human clinical trial to look at safety and preliminary efficacy, and are grateful for the first participant to have been dosed a few weeks ago. Because new genetic information is being introduced into the body, the way we do clinical trials for gene therapy is specialized. This first in-human study is small, about 8 people in the US to start. Safety is our top priority, so enrollment is staggered. We will wait several months before dosing the next people to ensure safety checks are met. We'll build the program over time. Should data support it, at a later part of the study would allow for more people to be enrolled. For more information on the study, you can visit the Huntington Study Group website. Finally, another approach looks to lower not only the mutant form of the protein. However, to be eligible for this type of drug, it requires individuals to carry a certain genetic variation, a specific SNP, on their HD gene. Working with our partner Ionis, who invented tominersen, we wanted to explore this approach in addition to other programs. We are currently running an epidemiology study to understand where and how many people with HD have the SNP we are trying to target. On the HD gene, there are many genetic variations, or SNPs, that are harmless and have no relevance to disease severity or progression. There are multiple SNPs that can be used as potential targets, guiding mechanisms for drug development. We developed a drug that targets a specific SNP and moved it through the animal testing phase. Now we need to understand the frequency and the geographic prevalence of our target SNP in the HD community. This is the purpose of our ongoing epidemiology study. Right now, we're screening volunteers in 8 countries, including 12 sites in the USA, to see who carries that SNP. If you're between the ages of 25 and 60 with very early or subtle symptoms, you may be eligible for a one-time blood draw to check. For more information, our trial information line can be contacted. I've summarized the status of the early program on this slide. If enough people carry the SNP, and if the lab findings continue to hold, we hope to launch a phase 1 study in the future. And now to close. I've told you about our comprehensive drug program efforts, but I want to also recognize how participation in research has continuing impact on HD science. At Roche Genentech, we are grateful for your contribution to our trials. We know your time and your data are precious, and that you participate in research not just for yourself, but for the next generation. Today, I share 2 examples of how we continue to commit to being a partner in the HD community. I mentioned the previous tominersen studies that stopped. Many of you in the room likely know someone who is involved with one of our studies and contribute to a tremendous amount of time and commitment. With that in mind, this year we've donated all placebo data from the Phase 3 Generation HD1 and our HD Natural History Study to the HD Research Consortium. We did this so other research efforts and future trials can benefit from access to this precious data. Thank you very much. The second example I want to share is about our digital app created for clinical research, trial research. This has accompanied most of our studies, and some of you may remember it if you participated in last year's HDSA Convention research pop-up. The app asks participants to complete various tasks like balance, tapping, and walking. The idea is to lead technology as a sensitive measure to help accelerate drug development. Based on all the data you help provide, we developed the HD Digital Motor Score that can be an official measurement in points in trials. This work was recently published, and Roche is partnering with other researchers to use the technology in studies. So please know that every time you raise your hand for research, you are leaving a lasting impact. So thank you. None of this would be possible without you. The families, the advocates, the investigators, and the staff who give your time, expertise, and even the thank you to your spinal fluid to move the science forward. Thank you very much. Thank you.
Anybody questions?
Well, just to let you know, I will be outside at the booth, so if you do have questions, I see one, great.
Hello, the question I have is the age limit. I would be excluded. The 25 to 60, I know is your choice right now. And why that limitation?
Yeah, no, it's a great question. We're really sensitive to that, and we understand that there are people, who maybe would be able to be great candidates for drug trials except for their age, and we want to keep all that in sort of in context to all the studies. Unfortunately, a lot of studies are made based on a lot of these criteria, and I think it's up to us to kind of revisit that and relook at that, but we do keep all that in mind.
Okay, thank you.
Mm-hmm. I know, I can't see it. Well, great. Thank you guys so much, and we really appreciate all you do.
Thanks, Rita. Appreciate it. And again, if you have additional questions or you don't necessarily feel comfortable speaking in front of a room full of people and everyone watching online, you can always catch Rita at the Roche Genentech booth out here afterwards. Next up, I would like to invite the team, well, here you are, the team from Change HD.
Thank you very much. Hi, good afternoon, everybody. It's very nice that we are situated where we are in this presentation because Change HD is different from the rest of the presentations you've been hearing. So, my name is Lori Jenish. I'm one of the team members from Change HD. My colleagues Kira Turkington and Robin are coordinators for two other studies that we'll talk about briefly at the end of this presentation. Change is kind of the umbrella study for what we're going to talk about and share today. Change HD is an observational study, and in that way, as people have been referencing, it is different from a clinical trial. We're not trying to change any outcomes or provide any kind of intervention in individuals. Instead, we're trying to enhance understanding over time of the disease progression. Our focus is particularly on early disease progression in younger people, as I'll talk about in a moment. We are a longitudinal study. That's a little tricky to talk about because we're in the last phase of a 7-year study. The study winds down in August of 2026, and so we so appreciate being able to be here today and have a chance to talk with people who may not be familiar with our study about participation, because what we have learned from our statisticians and from early data analysis, our baseline visits are especially valuable. So Anyway, thank you all for your attention and interest today. The research that we do is focused on, as I said, early brain development in persons with Huntington's disease. We're doing that in two ways. We're looking at brain structure by doing an MRI, and that way we are different from Enroll. Sometimes we compare ourselves to Enroll, but we do have an MRI imaging component. And with that, we're looking at brain structure, things that people have referenced already. We're looking at volumes, we're looking at cellular connections. We also have a functional component. How does the brain function in 3 areas? How does it function in motor skills, muscle tone, fine motor skills, balance? We do use the Unified Huntington's Disease Rating Scale motor component for one of our instruments. We also are looking at cognitive skills. Because we work with children, we play a lot of games on the iPads to do that, and it's fun and playful. But in terms of cognitive skills, we're looking at things like verbal memory, auditory memory, sequencing, pattern recognition, those sorts of things. Again, a lot of similarities to if you've participated in Enroll, the kinds of things you may have done there. And finally, we're looking at behavioral experiences and symptoms. And we do that through surveys for both the participant themselves and a proxy who they identify. Behavior of interest is things like sleep, family relationships, social relationships, hobbies, work, employment, school history. We ask some questions about drug and alcohol use, those sorts of things. People are always free to say, I choose not to answer that question. Change HD was actually, it's going into its 20th year, began at the University of Iowa, that's where Robin and I are from, as a single site study in 2006, as a pilot study funded in 2008 by NIH. It was called Kids HD at that time, and again, there may be people in the room who recognize Kids HD. It existed as Kids HD through 2019. We got promising results, published some from that, and we're able to write another grant. None of this is me, of course, but our principal investigator was able to write another grant and to involve 5 other universities in a multi-site research study. So we are growing the number of participants we have. We are almost up to 500 participants at this point in time. Our goals with Change HD are to replicate some of the previous study findings, which are that while Huntington's disease is considered a neurodegenerative disease, and of course it is, there may be some early benefits to the mutant Huntington gene, and seeking to understand that more. And we don't have time today to talk enough about that, to go into detail, but also to understand the pathophysiology of the developing brain in young people. So again, observational research and understanding, and to inform clinical research focused on preventative gene therapies, when are those best used for intervention, and also biomarkers. We have an NFL component of our study. As I mentioned, we're multi-site. These are where our sites are located. The University of Iowa is the lead site. We also have locations at the Children's Hospital of Philadelphia, Columbia University in New York, Vanderbilt University in Nashville, University of Texas at Houston, and University of California at Davis. There is a Change team of principal investigators, imaging specialists, and research coordinators. All of us use the same protocols. We share MRI imaging data. All of this is being done as a collective process, and we're going to begin data analysis when the study closes at the end of August. So it's kind of bittersweet that we won't be seeing people returning on longitudinal visits, but we will be starting to get to look at the data. Eligibility for the study is this. We are looking for people between the ages of 6 and 30 who have a parent or a grandparent that has a diagnosis of HD. We consider this at risk for HD, even though that's not scientifically accurate. And so It's important to know, and we get this question a lot, that participants in the study, because we are talking about children and that can be sensitive, do not need to know their genetic status. If a participant is over 18 and has gone through genetic testing because that was their choice, we prefer not to know the results of that genetic testing and to be blinded as study participants. So again, the eligibility criteria is having those family HT relationships. Participants who come to sites to participate must, under the age of 18, must have a parent come with them. Actually, it's quite a family affair usually. We have typically, or often we have young adults come with sisters or siblings and make it a weekend that they get to spend together. We have cousins come from 2 different locations and participate in a study, and they do this, you know, for the 3 or 4 years that they come longitudinally. So it is something that research coordinators work with families to individualize, to create flexibly based on what scheduling allows, and that sort of thing. The last thing in terms of eligibility that's a consideration is that the participant, if they're in that younger age range or older, depending on what family circumstances are, must have an age-appropriate understanding of how Huntington's affects them personally. The supports for participation that we are glad to be able to offer are these. Participants are paid $150 stipend for their participation. We know that doesn't cover the time that they put into getting to the locations and spending a day in the research study, but we are able to pay for that. We also are able to pay for travel costs, hotel costs, and meals for the duration of that participation. We want this to be as much as possible at no cost to the families. The study is funded by the National Institute of Health and NINS. The study staff are great about helping with travel arrangements and finding what works best for families. What else? I already talked about arranging visits with siblings. We encourage the families to have fun. Our principal investigator and the study lead, Dr. Napolis, talks about the challenges HT families experience and how this can be both a chance to contribute to research, experience the satisfaction, the empowerment that comes of that, but also to be together as a family and to have fun. And that really is what we see. People come into the day for the first time maybe nervous or anxious. And again, keep in mind, I'm talking about kids. I'm talking about younger people because our population, it's ages 6 to 30. Nervous, and when we ask them later in the day, hey, was it what you thought it would be? They say, yeah, it kind of was, but it was a lot more fun, or it was a lot easier, or that sort of thing. So people generally leave tired, but smiling. A typical daily schedule looks like this. We start in the morning. It really looks like a school day or a work day. And I apologize if this is too simplistic, but we wanted it to really give you a sense of what this involves, because it is children. We begin the day by meeting. 5, thank you. Begin the day by meeting. We do the consenting process, which is really just making sure people understand their rights and understand what's going to happen. And then we collect physiological measures. A blood draw is done at all visits. It's done at the first visit to collect the CAG length. And we also do some physiological measures like head circumference, height, weight, and blood pressure. We also collect a urine sample in terms of the MRI. I wanted to just say a note on genetic testing and other results too. This is research that is managed very carefully according to the ethical standards of research. Its results are not shared with participants or with any others. And we recognize the sensitivity of the genetic information we're working with, and that's something that our institutional review boards, our teams are very, very, very careful with handling. So that's important. After we go through that piece, we do assessments and surveys. They're cognitive, motor, and behavioral, like I said. We break for lunch. This is always informal. It happens when people are ready to do so. We place the motor assessments and the MRI later in the afternoon because they're best placed there. They're not as hard as the cognitive assessments. Okay, so that's our change study. That's the umbrella study. And the umbrella hypothesis really is, what does early brain development, pre-manifest, pre-symptoms, look like so we can understand that to better understand the course of the disease? The juvenile onset Huntington's disease study is different from that. In some ways and alike in some ways. It's a study for which one of the eligibility requirements is that the participants must have a JOHD diagnosis. And it too is an observational study that has a lot of the same components that I've talked about. The objectives of the JOHD study are to look at the differences between JOHD and HD, to look at the symptom presentation, brain changes, overall disease progression, because that too is different from HD, and also to look at the optimal time points for interventions, because our current understanding is that disease progression may be compressed. The JOHD study includes people with a diagnosis of JOHD, ages 4 to 30, having that diagnosis, having received that diagnosis before age 21, and there are some exclusionary factors that really have to do with the nature of the disease. Being able to participate in the day of testing is something that we do a really complete interview with a parent or caregiver about. All right, oh, there's more. JOHD visits involve 2 consecutive days. We slow it down from change because we don't want it to be as taxing. We have 2 years of visits, and they are approximately a year apart. This study too winds down in August of 26. And it has the same visit components with slightly different cognitive testing than change. Finally, we have a new study that we are doing that's a pilot kind of exploratory study that I'm going to turn over to Robin.
That's okay. I'm going to get the cowbell, aren't I? Okay. Dr. Napolis, I'm Robin Eisenbach, and Dr. Napolis and our team at University of Iowa are excited to launch our new pilot study, Mini HD. This study is an expansion of the Change HD study, looking at brain structure and function, but during the early development of infant and toddlers that have parents or grandparents with HD. As with Change, we are hoping this study will help explain how the gene responsible for HD affects brain development, look for the earliest signs of the disease, in hopes of finding the best time for intervention with gene therapy during development. This study is a lot like Change HD, except it's only at the University of Iowa, and it's also more of a mini version. So the visit, even though it's similar, the visit is much shorter. We do just a cheek swab versus a blood draw. We're utilizing the newly launched NIH Baby Toolbox for cognitive assessment. The MRI is also much shorter. With this data, we're obtaining, we know the data is very important, but so is our family's experience. We really strive to have a positive experience for the family, and especially with infants and toddlers. We know that that's going to be, not easy travel. So we're really striving to have flexible, schedule with the infant and toddlers. We pay for travel, compensation for participation, meals, and you get the 2 hotels that we generally use are on our Hawkeye campus, are really, really nice, and try to make it a really nice visit for our families. We have outside, we have our colorful table, so please stop by for more questions or grab some of our nice pens, but also share your family stories with us. That has been really enjoyable and learning a lot while we've been here. So thank you. Mary, go ahead.
Thank you.
Thank you both for that, and I hope what we're hearing with studies like Change and the JOHC study and the mini study is there are research opportunities for those who might not be able to qualify for other studies too that are vitally important as we think about HD over the lifespan. So thank you both very much, and yeah, if you got questions, catch them outside. Last but not least, I'd like to invite David and Victor to stage from Unicure.
Good afternoon, everybody. We decided for our presentation to not regale you with more PowerPoint slides and do a more kind of casual conversation. We envisioned a fireside chat, but Eric did not allow us to bring fire up here.
It's hot enough outside.
Yeah, that's right. So just imagine a fireplace here as we have our conversation. My name's Victor Sung. I know many of you here. I'm a neurologist, a COE director. I just rolled off the HDSA Board of Trustees. It's great to be back at convention, not having to do all the Board of Trustee duties, just here to hang out with friends.
Hi, I'm David Margolin, also a neurologist and working at Unicure as Vice President heading clinical development, and I'm the program lead for the HD project. It's a pleasure and an honor to be here today.
I did want to add that I am a paid independent consultant for Unicure, and I'm a principal investigator in the AMT 130 trial that we will be discussing today, and that this session will be recorded and posted online. So as we talked about, we're here really today to talk about Unicure's trial studying AMT 130. David, can you go through what is that study and how does it work?
So AMT 130 is a gene therapy, as you've heard about from Spark and Roche. It's a candidate that's being investigated in people with HD. In HD, as you've heard, brain cells are thought to be damaged by the buildup of mutant Huntington, and AMT 130 is designed to reduce the production of the Huntington protein. It targets messenger RNA, which plays a role in how proteins get made. AMT 130, as a gene therapy, instructs cells to produce a special RNA that reduces production of the Huntington protein using a natural cellular mechanism called RNA interference.
As I mentioned, I'm a principal investigator in the AMT 130 trial. I also see patients in clinic, HD patients in clinic, and a number of my patients have participated in this trial. So I have some experience and can use that to go through a little bit about how this drug is administered. As Dr. Gandhi was explaining in the Spark presentation about how gene therapy is done, gene therapy and AMT 130 is a single surgery, and during that single brain surgery, AMT 130 is administered under general anesthesia, and the surgeon uses MRI guidance to guide the injection of the drug into this key part of the brain called the striatum. The striatum consists of two structures called the caudate and the putamen. If you've heard of that in all of your time with HD, the striatum is the particular part of the brain that we know is affected the most in HD. So when you have loss of that part of the brain, we can attribute many of the symptoms of HD to the striatum. Thus the selection of injecting the drug there, right? Once it's injected, the AMT 130 is designed to stay in the brain cells for the life of that brain cell. And over a lifetime, make these blocking RNAs that will decrease the production of Huntington protein over a lifetime. It's important to note that AMT 130, unlike maybe some other gene therapies that you may have read about in other diseases, it does not integrate into the DNA itself of the brain cells. So it's not changing your DNA code per se. David, can you go through a little bit the goals of the trial?
Sure. We're seeking to establish and learn about the safety and efficacy of AMT-130. We hope the data will show that it can delay, possibly prevent progression of the symptoms of HD, providing long-term benefit from that single treatment and helping patients maintain a quality of life. We initiated our first clinical study in 2019, and we follow each participant for 5 years. By 2024, last year, enough patients had reached the 2-year milestone that a picture began to emerge, suggesting a slowing of disease progression. And we reported at that time encouraging trends in the primary efficacy endpoint of the composite Unified Huntington's Disease Rating Scale, the CUHDRS for short, which provides a single number, a single score that represents a combination of different measurements covering a range of HD symptoms, cognitive, motor, and function. But in the study, we've also learned that AMT-130 has serious risks, but so far it appears that it's generally well tolerated and that we believe the risks are manageable. We've not yet proven that it's safe and effective. It's not yet approved by FDA or any regulatory authority, and the study is ongoing. Data continues to be generated and analyzed.
David, you mentioned that there was a major update. So there was a significant data update released in July of 2024, just about a year ago from now. Do you want to highlight any particular things that were part of that? But that was after last convention, right? So anything from that data update that you want to highlight for us today?
Yeah, the CUHDRS showed a slowing relative to the external comparator group, which was a natural history cohort. And I echo what Jane had said about the value that the companies perceive in these natural history studies like Enroll, and encourage you all to continue to participate. And we're studying 2 different dose levels, and both of them seem to show some benefit relative to that external comparator group.
As someone who treats HD patients, like many of you every day, I feel the pressure, like many of you do, like many of us in the room do, to have the first across the line, right, to be FDA approved as disease-modifying therapy for HD. I think we're all aware we don't have one yet. There's a lot of pressure to get that approved. As we consider all the candidates, as we've been discussing today, given the invasiveness of the brain surgery that would be required for something like a gene therapy, I've certainly heard from my patients the importance of having it be safe, having it be FDA approved or at least conditionally improved before they would consider something as invasive as a brain surgery. So, and we've talked about some of the regulatory bodies. David, can you share how uniQure has been engaging with the FDA and what their response has been to the data that you've presented to them?
Yeah, we've had a series of meetings with the FDA, and frankly, we've been very encouraged by their interest in supporting development of treatments for HD. The FDA has told us that with regard to our program, that the ongoing study that I discussed earlier can be compared with natural history data as the primary basis for our licensing application under what they call the accelerated approval pathway. And no additional study should be needed to gain a licensing approval, or at least for their decision. That accelerates by several years the time frame in which AMT-130 might become broadly available to US patients.
That's really exciting. David, can you tell us what is coming up for uniQure for the rest of 2025?
Well, we've previously announced we completed enrollment in an additional cohort in the current study, and we continue to collect data on all the participants in this study. We're now busily preparing to examine results from the 3-year follow-up and expect to submit a licensing application to the FDA probably in early 2026. And we'll also be providing the public with an update on the study results later this year.
It's exciting. I wanted to take this opportunity to thank all of the brave people that have participated in this cutting-edge research, like the AMT 130 trial. It takes a lot of bravery to do things that have not been proven so far. And then for all of the participants, like many of you in all of the research trial, we couldn't do without you. So can we get a round of applause for all of our brave participants in research? We do it for you, we couldn't do it without you. Something that I've heard from my patients that have participated in this trial and many other trials is this notion that all of you guys have, which is that even if participation in this trial does not lead to an approved drug, that the participation in the trial could lead to things that help the community in the future, right? And the participation in clinical research is a way of finding hope. It's a way of having a legacy that you leave behind. So I think just it's amazing what you guys do in your participation in clinical research. So just an encouragement to all of you guys to continue to fight the fight and keep that hope alive. Related to that, David, how do you think the community, the HD community, can stay engaged and involved with everything going on in research, and in particular, unicure's next steps?
Yeah, well, I'm proud to say that uniCure has been very much involved with patient organizations through several years now, including HDSA, HDYO, and others. We've convened, I think, 4 now different patient advisory boards. The first one even in 2018 before the study began, and it helped shape the design of our study. We wouldn't be where we are today without the involvement and contributions of the HD patient community. So we do appreciate both participation in the studies and in supporting the research generally. For that, we thank you. But for the future, it is an exciting time, not only for our program and the prospects of a potential regulatory review and approval, but from the other companies that you've heard about from the other speakers today. There's really hope, I believe, for effective treatments. But becoming involved in these research projects is something that everyone in an HD family can do. I encourage you to do that, and also to get before the regulators, the lawmakers, and share your stories. Let them, help them to educate them on the reality of living with HD and the urgent need for effective treatments.
In closing, I want to thank David for being here with me today, for candidly discussing where things stand with unicure's AMT 130 program. I think hopefully you guys all agree with us that this is an exciting time in research in HD, and there's some exciting updates with the AMT 130 program. For me, this falls under this umbrella of maybe cautious optimism, which certainly in the wake of failures that we've had in HD trials over many years, I will certainly take a position of cautious optimism. I want to thank uniCure for their support and engagement with the HD community. Certainly thanks to HDSA for hosting this event here. I hope you guys have had a great time here at convention. A round of applause for HDSA. And thanks to all of you, as we said, for participating in clinical research, for engaging with all of us who do the clinical research. As I said, we do it for you, and you inspire us as much as hopefully the research inspires you. If you have questions about unicure's program, you can find them at the booth, which is right out those doors, and uniCure representatives will be happy to field any questions there. We're almost out of time, and I'm very sensitive to coming to the end of a 90-minute session. I know, some of you are getting a little sleepy and ready for nap time. But in general, stay tuned for exciting announcements for all of the programs that have been mentioned today. There are exciting times ahead in HD clinical research. Keep the hope alive. Thanks so much, everybody.
Thanks, Victor and David, and thanks to all of our presenters. Please, another round of applause for everyone who presented this afternoon.